Good day everyone, I am Kyla Seria from BS Enter 1-2. Today I will discuss the gene transfer and the selection using marker gene. First, I will discuss the gene transfer. What is gene transfer? Gene transfer is the mechanism by which DNA is transferred from a donor to a recipient. Once donor DNA is inside the recipient, crossing over can occur. The result is a combining cell that has a genome different from either the donor or the recipient. The first gene transfer experiments with plants took place in the early 1980s. The transferred gene is known as transgene, and the organism that developed after a successful gene transfer is known as transgenic. Transgenic plants have been obtained by using agrobacterium-mediated DNA transfer and direct DNA transfer, the latter including methods such as particle bombardment, electroporation, and polyethylene glycol permeabilization. The majority of plants have been transformed using agrobacterium-mediated transformation. Gene transfer has three methods. The first one is agrobacterium-mediated DNA transfer. It is the most popular methods. It involves the natural gene transfer system resident in the bacterial plant pathogens of the genus Agrobacterium. It invades many dicot plants when they are injured at the cell level and causes crown gall disease. The ability to cause crown gall disease is associated with the presence of the TI or tumor-inducing plasmid within the bacterial cell. TI plasmid can be used to transport new genes into plant cells. The TI plasmid is a remarkable features of the TI plasmid is that after infection, part of the molecule is integrated into the plant chromosomal DNA. This segment, called the tDNA, between 15 and 30 kb in size, depending on the stain. tDNA contains 8 or so genes that are expressed in the plant cell and are responsible for the cancerous properties of the transformed cell. These genes also direct synthesis of unusual compounds called opines that the bacteria use as nutrient. The VI or virulence region of the TI plasmid contains the genes required for the tDNA transfer process. The genes in this region encode the DNA processing enzymes required for excision, transfer, and integration of the tDNA segment. TI plasmid-mediated transfer of gene into a plant. The TI plasmid has innate ability to transmit bacterial DNA into plant cells. The gene of a donor organism can be introduced into the TI plasmid at the tDNA region. This plasmid now becomes a recombinant plasmid. By agrobacterium infection, the donor genes can transfer from the recombinant TI plasmid and integrated into the genotype of the host plant. I will show you the gene transfer in plants using TI plasmid. One naturally occurring mechanism of gene transfer to plants is via the plant pathogen Agrobacterium tumefaciens. This bacterium transfers some DNA referred to as tDNA from a plasmid called the TI plasmid, so that the genes causing tumor formation have been eliminated, but the tDNA is still transferred to plant cells. If a gene of interest is inserted into the tDNA, it will also be transferred to a plant cell. The recombinant TI plasmid is then transferred into an agrobacterium cell lacking a plasmid, generating a bacterial cell containing the recombinant TI plasmid. This bacterium can then attach to a plant cell to begin the process of gene transfer into the chromosome of the plant cell. The tDNA is replicated within Agrobacterium tumefaciens and only a copy of the tDNA is transferred to the plant cell. This recombinant tDNA is incorporated into a plant chromosome. Using the appropriate growth hormones, this cell may continue to divide and thereby generate an entire plant that carries the gene of interest. In this way, it is possible to produce a transgenic plant that carries a cloned gene. The next method is particle bombardment. Particle bombardment is an alternative method that is suitable and efficient for many plant species. It facilitates DNA delivery into the intact plant cells through simultaneous multiple gene transfers with no biological constraints or host limitations. It also employed for DNA delivery in transient gene expression studies to investigate the plant gene expression and for its ability to introduce DNA directly into different tissues. Different plant organ tissues have been used as bombardment targets: pollen grains, immature embryos, embryogenic colluses, embryogenic suspensions, hypocotyls, flowers, fruits, and root hairs. This technique is generally less efficient than agrobacterium-mediated transformation, but has nevertheless been particularly useful in several plant species, most notably in cereal crops. And the third method is microinjection. It uses a small needle to insert the gene into the plant cells. It is the process of transferring genetic materials into a living cell using glass micropipettes or metal microinjection needles. Glass micropipettes can be of various sizes with tip diameters ranging from 0.1 to 10. DNA and RNA is injected directly into the cell's nucleus. In the next slide, I will discuss the selection of using marker genes. In a transformation experiment, the proportion of transformed cells is usually small compared to the number of cells which remain unaltered. In order to select only cells which have actually incorporated the new genes coding for the desired trait are fused to a gene which allows selection of transformed cells, so-called marker gene. The expression of the marker gene enables the transgenic cells to grow in the presence of a selective agent, usually an antibiotic or a herbicide, while cells without the marker gene die. The first marker is a bacterial aminoglycoside phosphotransferase gene. This is one of the most commonly used marker genes, also referred to as neomycin phosphotransferase 1,1. This gene codes for an enzyme which inactivates the antibiotics kanamycin, neomycin, and G418 through phosphorylation. In addition to NPT2, a number of other antibiotic-resistant genes have been used as selective markers. Hygromycin phosphotransferase gene conferring resistance to hygromycin. Next one is the herbicide tolerance gene, another group of selective markers. It has been obtained through the incorporation and expression of a gene which either detoxifies the herbicide in a similar manner as the antibiotic-resistant gene products, or a gene that expresses a product which acts like the herbicide target but is not affected by the herbicide. Herbicide tolerance may not only serve as a trait useful for selection in the development of transgenic plants, but also has some commercial interest. Herbicide tolerant transgenic plants are therefore among the first crops approaching market introduction. Some related terminologies in biotechnology. The first one is the biotechnology, the controlled use of biological agents such as microorganisms or cellular components for beneficial use. Next is the antibiotechnology, pertains to all such activities other than the conventional approaches that either improving the genetic makeup, phenotypic performance, or multiplication rates of economic plants, exploiting plant cells or cell constituents for generating useful products and or services. Next is the genetic engineering. Genetic engineering or recombinant DNA technology is the technique of moving genes from one organism to another or putting them back in different combination to produce an improved organism. Next is transgenic plants or GMO. Transgenic plants are produced by means of genetic engineering. They contain genetic materials from other organisms like other plants, animals, viruses, bacteria, or fungi, or genetic materials synthesized in the laboratory. Next is the Bacillus thuringiensis, or BT. Is a bacterium which kills only certain insects or lepidopterans but not other organisms. A gene from a BT is used to produce a protein or delta endotoxin that kills certain insect pests or lepidopterans such as a corn borer. BT corn. BT corn refers to a variety of corn where the BT gene has been inserted. This makes corn resistant to the Asiatic corn borer, the most prevalent insect pest of corn in the Philippines. Next one is the organogenesis. It is production of various organs, roots, shoots, etc. from cells and tissue culture. Next is colus. The cells on an agar medium develop into an organized mass. Next is the classification of tissue culture techniques. One is the embryo culture. Embryo culture, young embryos are removed from the developing seeds and are placed on a suitable nutrient medium to obtain seedlings. The application of embryo culture are recovery of distant hybrids, recovery of haploid plants from interspecific crosses, recovery of crosses, shortening the breeding cycle, and overcoming dormancy. The next one is meristem culture. Meristem culture is the cultivation of axillary or apical meristems, particularly shoot apical meristem. And the application of meristem culture are micropropagation, recovery of virus-free stocks, germplasm exchange, and germplasm conservation. Next is the anther or pollen culture. Growing of haploid plants from pollen grains or anthers on a suitable culture medium. The application of anther or pollen culture are useful in obtaining haploid plants, production of homozygous double haploid. Next is tissue and cell cultures. The application of tissue and cell cultures is biochemical production and biotransformation, clonal propagation, semiclonal variation, somatic hybridization. Genetic transformation, organelle transfer, and germplasm conservation. In the next slide, I will show you the list of transgenic crops developed or under development taken from the fundamentals of crop and science. The first crops is the canola. The traits transformed in canola are herbicide tolerance, improved disease resistance, and high lauric acid. The next crops is corn. Traits transformed in corn are control of Asian corn borer, herbicide tolerance, improved quality like high starch, high lysine, improved protein. Next crops is cotton. Traits transformed in cotton are control of boll weevil, herbicide resistance, improved fiber quality. Next crops is potato. Traits transformed from potato are resistance to Colorado beetle, multiple viruses resistance, and higher starch. Next crops is rice. Traits transformed in rice are high beta-carotene content or golden rice, wider adaptability salt or saltol, and submergence or submarina tolerance, improve hybrid technology, herbicide tolerance, resistance to bacterial blight, and resistance to stem borer. Next crops is soybean. Traits transformed in soybean are herbicide tolerance, modified high oleic acid, insect resistance, and virus resistance. Next crops is vegetable, fruits, and ornamentals. Traits transformed in vegetable, fruits, and ornamentals are virus resistance or PRSB, tomato, insects, insect resistance or BT eggplant and potato, and delayed ripening in papaya and tomato. That's all. Thank you.